Welcome to the Neuro-Bio podcast with me, Baroness Susan Greenfield. I'm CEO and founder of Neuro-Bio, and I'm joined by Dr. Richard Mohs, who is the Chief Scientific Officer of the Global Alzheimer Platform and co-author of over 350 scientific papers, and by Dr. Mike Zurawski, who's the Chairman of Neuro-Bio. This series of discussions focuses on new insights resulting from the current research into Alzheimer's disease, and hence to the revolutionary work Zurawski and Neuro-Bio are envisaging to bring pre-symptomatic testing and an effective cure to the market. Alzheimer's is an extremely pervasive and severe disease with more than 55 million people living with the condition worldwide and 10 million new cases each year. That is why Zurawski and Neuro-Bio are committed to at last putting a stop to its devastation. Zurawski, as the first corporate finance house focused solely on impact for both the climate and people, is honoured to be working with us on this project and to continue the journey into disrupting the corporate finance sector. Join us now in exploring behind this tragic disease and in learning more about Neuro-Bio's work and Zurawski's 10 million fundraise to bring its innovation into the mainstream.
So thank you, Susan, for the introduction. I think this is a good time to address this initial question about where do we stand now with research onto new treatments for Alzheimer's disease. The past few years have been very active in the field. As many of your listeners probably know, there's been a tremendous amount of effort devoted to trying to understand what role amyloid beta, that is the protein that goes into forming the amyloid plaques in the brains of patients with Alzheimer's disease, might play in the development of clinical symptoms for this disease. And over many years, a lot of different approaches were tried to reduce the quantity of soluble monomers of the amyloid beta protein with secretase inhibitors and some antibodies directed at soluble monomers. And unfortunately, none of those approaches had any clinical benefit. In the past couple of years, focus has shifted in that realm, at least, to antibodies that are designed to remove aggregated species of the amyloid beta protein. And we have now several that are in development: lecanemab, donanemab, aducanumab. And those molecules are quite potent at removing amyloid plaque. The availability of biomarkers for brain amyloid has enabled us to show, or enabled the field to show collectively, that those kinds of antibodies are quite effective at removing amyloid plaques. And we now have some indication that that approach has at least some clinical benefit. We have two, at least in the US, that have been given accelerated approval for clinical use based on their effect to reduce amyloid plaque. And the data from lecanemab, that's the ASI antibody, have been submitted for regular approval. So that's quite promising, and it shows, I think, the interplay between the development of biomarkers and therapeutics, and how those two, if they're done hand in hand, they can speed the development of therapeutics. I think it's quite likely that lecanemab will be approved, and maybe some follow-on products, donanemab and so forth, for slowing the progression of Alzheimer's disease, because they do seem to have a clinical benefit when administered in patients, at least at the early stages of the disease. But we've also learned that their effects are modest. The studies with lecanemab and donanemab are quite large, so we have a pretty good estimate of the magnitude of the effect size with those compounds, and they seem to slow the rate of progression by about 30%. Which is, of course, very real and highly desirable, since we haven't had any new treatments for Alzheimer's disease in the past 26 years. I guess nothing has been approved. So that's very promising, and I'm sure more progress will be made there. But it also shows some of the limitations of that approach. These approaches do not restore patients to their previous level of functioning, and do not stop the disease entirely. And I think that there's growing recognition that full treatment for this disease will require multiple approaches. There's other biological abnormalities associated with this disease, and that it's fruitful to pursue those other approaches so that we can do a much better job than just slowing by 30%. I think the other thing that's really been quite dramatic in the past few years is the tremendous progress we've made on blood-based and other fluid-based biomarkers. Of course, the development of PET imaging technology has allowed us to envision in life some of the pathologic features of Alzheimer's disease, and that, of course, is very helpful. We can look at tau tangles and amyloid plaques, but that technology is quite expensive and quite invasive and difficult for patients to use. And so there's been a much greater emphasis in the past couple of years on things that can be measured in blood or saliva. There's some work on skin biopsies, but obviously blood-based biomarkers are the kind of things that physicians are typically used to using in other diseases. And the fact that we can measure amyloid beta and tau and neurofilament and now T14 in blood is a great advance, both for enabling greater precision in clinical trials and also for help in the clinical evaluation of patients. So I think after long struggles, I think we're starting to make some progress, but we obviously have a long way to go, and there's a need to look at a lot of different approaches to treating this disease before we can fully expect patients to have a high quality of life, even if they're at risk for the disease.
Thanks, Richard. And let's move on as a perfect segue into alternative approaches, because as you rightly say, antibodies to amyloid do have some effect in slowing progression, but I don't think anyone would say that they are giving an effective treatment or cure of the type that we would welcome, where you're stopping any more brain cells dying. And I think what's missing is that for two reasons, there's something missing here. One is even before you present with memory problems, there is a 10 to 20 year time lag. So we know that the slow process of cell death, this gradual process of cell death in the brain, that can occur 10 to 20 years before you have any signs that there's a problem. So obviously it means that if you only treat someone when they present with those signs, you're closing the door after the horse has bolted. You've lost 10 or 20 years. And there's an urgent need, therefore, to have some kind of indication, some screen that would be available to the whole population, to us three, to you listening, that could be applied so that you could see that there was already the process underway. Tied in with that is the other problem, is that to the best of my knowledge at least, we're the only approach, Neuro-Bio is the only approach that focuses on the rather interesting feature that not all brain cells are created equal, that some are much more vulnerable than others. And the ones that are primarily vulnerable are deep down in the brain. They come from a different part of the embryo, which means they might have very different properties. Interestingly, in the early stages of neurodegeneration, before the symptoms, although there's cell loss, you don't get amyloid. So although amyloid is a very obvious and appropriate target, as Richard has outlined, for the later stage, in the very early stages of the degenerative process, it's not obviously there. So obviously it can't be the primary target. So obviously that's not going to eventually give you an ideal therapy. And what we have done is to explore these very basic cells in the hub, the ones that come from a different part of the embryo, to find out what is special about them. And they may not have amyloid, but they do have other things going wrong. And one of the things they do have that Richard just mentioned is a much less known chemical. We call it T14 because it consists of 14 little building blocks, so-called amino acids. And we've been working with this molecule for the last 20 years and have increasing evidence that it promotes cell growth in the healthy body when it's developing. But if that's inappropriately activated, it's like Jekyll and Hyde. And if it's inappropriately activated and it's there in these very vulnerable cells, then in the context of the adult brain, it will have the opposite effect to what you want. It won't be Jekyll, it will be Hyde. It will go from being a benign developmental agent to actually being toxic. But of course it doesn't know that, so the more that happens, the more the cells will try and compensate by releasing more of the evil T14, and the more you'll have this cycle of perpetuating neurodegeneration. So why we're different, what we're filling in here is an alternative to amyloid, not so much that it's an opposing view, but it's upstream of a process that will subsequently, later on, in other brain areas, lead to amyloid. And those other brain areas are the ones that show problems with memory. The very basic cells in the brain aren't related to memory at all. They're related to arousal and sleep-wake. They're very basic. Therefore, for those first 10 or 20 years, if you see anything at all as a problem, what you'll see are sleep disturbances or mood disturbances. You won't see so-called cognitive impairment. And that's much harder things to light on. If you just have a sleep disturbance, that's much harder to think you've got Alzheimer's. So really, what we need now is to devise a marker for that stage, either in saliva, as Richard mentioned, or nasal swab for what Americans call an antigen test. We in England call a lateral flow test. If we could do that, then you could do that routinely, and that would really be a game changer because you'll detect there's a problem underway before you had any cognitive symptoms at all. And if you had a drug that worked, that intercepted it, then you'd never get the symptoms, and that would be an effective cure. So that's what at Neuro-Bio we do, and it's based on this basic process we've discovered in the primarily vulnerable cells that is only now starting to get, I think, traction. In order to intercept it, we actually have a drug, and the drug is the same T14, but it's literally modified, it's literally bent out of shape so that it occupies the same place that the naturally occurring toxin would want to sit on. But because it displaces it, and it itself is inert, it's a bit like having your hands tied behind your back. So no one can sit in your seat and can do anything, but you can't do anything either because your hands are tied behind your back. This drug, which we call neurobiopeptide 14, NBP14, seems to successfully displace the bad T14. And so far, we've shown in an animal model of Alzheimer's that it seems to be effective. The animals don't develop the memory problems that you see in the so-called controls, the animals treated with saline. So where we're at the moment is quite exciting because what we think we've hit on is a very basic process for cell growth that's inappropriately activated. And we describe Alzheimer's, our explanation is that it's an aberrant triggering of a normal developmental process in the wrong context. And because it's in the wrong context, it's toxic. But it does mean that if we can devise a way of detecting it before there are symptoms in saliva or blood possibly or nasal swabs, we already know in the brain it is elevated at the pre-symptomatic stage. So all we have to do is reproduce that profile in some kind of easily accessible tissue and then eventually give our drug, which is the bent out of shape molecule. And hopefully, if humans are like mice, and that's a big stretch of imagination, then that could be offer a prospect of an effective treatment. Mike?
Absolutely. So I'll think I follow on after the science with a little bit of the finances to really illustrate the scale of the issue, the scale of the challenge, and the scale of the opportunity. And obviously, the monetary value isn't the best segue to understand the scale of something that is so personal to so many families in terms of impact. But I do think it's still illustrated quite dramatically. And currently, as of last year, the global cost of Alzheimer's and dementia was about $3 trillion. To put it into context, the entirety of the economic output of the UK is around $3 trillion, which is just incredible to think about. And I think that really hits home as to just what this problem is in terms of impact. And that's not the end of the story. We know we have an aging population, which means that more and more people will fall into this disease category. And in the next 10 years, we expect that to double in terms of impact and monetary loss. That's to say that it will be about $6 trillion, and then another 10 years after that, it'll be about $10 trillion. At that point, we're talking about the combined, again, economic output of France, Germany, and the UK put together. Or to put into another context, the entirety of Latin America and the Caribbean, and a few, I'd say about half of the EU. So I think that's very important to just keep in mind as we talk about obviously societal impact here. And that's actually vastly underestimated, and that's widely accepted that we don't really understand fully the impact of this disease and that the effect it has on the families. because of the fact that a lot of families, especially in the very early stages of the disease, actually shoulder the burden economically by themselves without necessarily being able to tap into specialist care and health systems. The numbers I just gave are actually based mostly on professional caregivers and health systems and therapeutics. They don't really count very well the shadow impact on the wider economy. So that's actually underestimated costs. And I think to build on that and just to sort of put it into context of Neuro-Bio and what we are trying to achieve here, the entirety of that market is potentially up for grabs by Neuro-Bio. And that's to say, Neuro-Bio isn't looking to necessarily just move the needle a couple of percentage points in the right direction. And it's not just trying to find one solution, one silver bullet to everything. The real beauty here is the fact that we're working on a diagnostic, a truly pre-symptomatic diagnostic product, which will be able to tell patients that they are at an increased risk of developing the disease, and then also a therapeutic. So you're able to treat your patients before they develop symptoms, before they need any kind of care, be that from their family and social circles and the wider health system. So I think, just to, again, illustrate that with numbers, because that's sort of why I'm here. The first 5 years of just a diagnostic, in terms of revenue, that's hundreds of millions of dollars in revenue. And I use that not as a measure necessarily of the commercial, which is obviously still important as a company, but in terms of just the sheer need of patients. The amounts in financial terms are emblematic of just the need for something like this to patients and health systems. And obviously the therapeutic, once that's in the market and post-clinical trials, we're talking about billions of dollars in the first 5 years of sales post approval. And obviously that's a long way away from now, but we're actually much closer than people, I think, realize. Now that we're looking forward to our first clinical trials, it's really a hugely exciting time.
So we're obviously biased in our excitement here because I feel that it's like a jigsaw puzzle, that every time we get a new observation, a new result, it completes the picture even more. People often ask, well, just how revolutionary is this? Is it like Viagra? Is it like aspirin? Is it that? And I think it's different from both of those, because Viagra, as far as I know, was discovered by chance, by serendipity, while exploring something else. Its application was for something else. Aspirin, I think people took a long time before they worked out how it worked, but they knew it did. We're different in both those cases, but I think it could be equally outreach in terms of the effects it could have. In that we started with a single story that I developed at university when I had a chance to look out the window and read lots of literature. And as you do when you're an academic, you join up the dots and try and tell a story. And if the story doesn't fit with the facts, you reject the story and devise another one. That's one of the luxuries that you have when you're teaching very bright people, as I was. You're in constant dialogue and you're held up to constant scrutiny and accounting for trying to get a really true explanation, not just one that will get you to the next milestone, but a really true explanation. And that's what I did before we started Neuro-Bio. And in that sense, I think we are very special, and that it does come from a real narrative that is backed up by the literature, by known clinical facts. And actually, more than at the time when it was first thought of, we can now see that this process that we're targeting and manipulating and that we want to see reflected in a pre-symptomatic screen and intercepted with the drug, it actually applies way more generally to the body. Because if you say neurodegeneration is an inappropriate form of development, that is, of course, one of the definitions of cancer. So we do have indeed an oncology application that seems to be being validated. Similarly, we know there's a part of your body that's in constant development, and that shares the same origins as your brain cells, and that's your skin cells. And there's increasing evidence linking disorders in the skin with neurodegenerative disorders. And again, we are showing that our T14, our chemical, seems to be at work in the cells of the epidermis of the skin. So there's a dermatology application as well. So whilst it is, I think, revolutionary, it's not like Viagra or aspirin in that it actually is uncovering a very basic process. How we modify it, how we intercept it, what disorders we apply to are very wide, and we're still discovering them. But our main focus, of course, is Alzheimer's. And in that sense, I hope it will be revolutionary because I think we're the first to show a pre-symptomatic elevation in T14 in the brain, which I don't think anyone else has, and also because it is so effective in the animal model. So, of course, I'm biased, but someone like Richard, of course, who's had a ringside seat, many of such claims, I'm going to hand over because I'm sure he probably can give a more objective and less biased appraisal than I can.
Well, look, Susan, it's interesting that you bring up why Neuro-Bio is unique, and I think it really is, and it has some characteristics associated with the program that you've developed that really make it extraordinarily promising and unique. You mentioned earlier that there is a relationship between T14 and specific vulnerable cell groups in the brains of patients with Alzheimer's disease, and that's certainly a link that has been missing between the other pathologic features of AD, the amyloid, which is kind of diffusely, it's sort of throughout the brain, doesn't really have much in the way of specificity and doesn't link up very well with what we know to be the selectively vulnerable neuronal groups. And that is a deficiency that has been not shared with the Neuro-Bio approach. You've really linked up the specific neurons that are vulnerable with the pathology that you're aiming for. The other thing that I think is unique is the link to developmental processes. Obviously, age is the most significant risk factor for Alzheimer's disease. I mean, we don't get typical Alzheimer's disease early on in life. It's something about the aging process. And what you've done is identified a neurodevelopmental process that may, as you note, have gone awry in late life in some people, and that leads to selective vulnerability of these neurons. I think that the combination development of a unique and different biomarker, which could be readily available and scaled up for large numbers of people, very different, say, than PET scans can. Anything that can be measured in blood or saliva is easily scalable, presumably. You can even do it without people showing up at the clinic. I mean, there's things that people can do now. You just send them a kit at home and they do a cheek swab or something like that. And if you can develop a biomarker like that, you could actually have a screening process that doesn't even require people to go into a clinic. They might if the test comes back positive for counseling and so forth. So I think that there's a number of things about the Neuro-Bio program that are really extremely promising and complementary to what's going on in other realms. And I think it has the potential to really have a very scalable impact on a disease that, as Michael mentioned, is a very common disease. This is not one of these rare genetic forms of cancer where there's only a few thousand of them in the world. And obviously for those people, it's a very devastating disease. But this is a common disease that affects all societies. And with the advances in other parts of the healthcare system where we have large pools of older folks in almost every country in the world, diseases like Alzheimer's disease are the public health problem of the future. And we need approaches to those diseases that are patient-friendly, economically viable, and scalable to large populations. So I think the things that Neuro-Bio is doing, both with the development of scalable biomarkers and therapeutic approaches that could be applied to people who are at the at-risk stage of disease, this is tremendously promising and very likely to have a major impact on the field.
Let's hope. It seems like a long journey. When I look back to how I started, which was very unorthodox in that I initially didn't do sciences at all. I hated science at school. And I actually did entrance for Oxford on Latin and Greek and ancient history and maths. So I came up to do what's called classics. And had someone said, you're going to end up as the CEO of a biotech, I would have thought they were completely batty. But I think it's that unorthodox journey that has led to an unorthodox approach and recognition that just such a disruptive approach is needed. I think if you don't know what you don't know, then you can just bash on and you can ask questions that might be like the emperor's new clothes and actually come to terms with things and get to the basics in a way perhaps that people that make assumptions derived from dogma don't make so well. I feel very privileged in that it has been a very interesting and exciting journey. And during COVID, that would be one particular chapter, for example, when, like everyone in the world, one stared into the abyss and wondered what was going to happen to one's life and one's friends and family and so on. Actually, for Neuro-Bio, it was an amazing time because we outsourced the work for testing in animal models to Italy, of all places, which was in Europe, the epicenter initially of COVID, but they were allowed to stay open, the company, because they were a medical lab. And whilst we were closed down and making people redundant and having people on furlough and therefore not as productive as we could have dreamt of being, this lab actually did the first of the animal testing to see if our drug actually worked. And there was no reason, we just were guessing. We were guessing the dosing protocols, we were guessing the dose, we were guessing the test, we were guessing the model, because we had no benchmark to go on. And against all the odds, it actually worked. We got the right dosing protocol and the right animal model and the right results that we published about just under a year ago now. So in a sense, COVID wasn't so much of a problem as it was to many companies. And from a financial side, we were also saved by the British government having an investment scheme called the Future Fund, which matched for new investment pound for pound. They came in as investors, but nonetheless, it doubled the investment without very little due diligence. And that was truly a lifesaver. And we're very grateful to the government for doing that. So in that sense, we survived COVID, but we actually did better than merely survive. We actually flourished. And now we're at a time when we are thinking about these new treatments. And what we're attempting to do, our first so-called clinical trial, which we hope to piggyback on the back of existing clinical trials, is a very simple idea. It's to measure T14, the chemical we think is the driver, to measure it in saliva. Now, in a very preliminary experiment, we showed that the T14 in saliva within our research group, it actually seemed to be related to age. And as befits a molecule that promotes normal cell growth and renewal, in normal aging, it starts to go off. And unfortunately for Neuro-Bio, there was only one person aged over 45, and that was me. And mine was very low. But it was sufficiently informal but indicative that we've now teamed up with a company who are regulators. We've teamed up with the Alzheimer charities in the UK, and we've teamed up with an institute near Oxford, we hope, that has a very fast way of measuring peptides without using antibodies. And with a fair wind and a goodwill, we're hoping to combine all these technologies and stakeholders so that we can, within the next few months, see indeed is T14 elevated in people with diagnosed Alzheimer's compared to controls. And if it is, that will then lead to the development of an antigen test, a lateral flow test, where you could see how your T14 relates just to your age. And if it was deviant from that, then you'd go to the doctor and have a more detailed quantifiable test to see indeed if the problem was already underway. And if it was, then you could enroll in clinical trials that are already existing, or eventually in a few years' time, hopefully our drug would be available. And that will be an intranasally administered drug. That seems to be the best route. And in the experiment, we did it twice a week, so it wouldn't necessarily be every day. But it'd be able to be administered at home. You wouldn't have to go into a hospital like you do with many of these antibodies. It wouldn't be a whole day in a stressful alien environment. You could have it at home, and it would be relatively cheap to do, and a lot would get into the brain because it's a much smaller molecule. So the dream scenario would be, first of all, an off-the-shelf at the pharmacy, you just, like you might have done for COVID, you do a lateral flow test, antigen test. If you are outside of the expected age range, you then go to your doctor. You then have, with all the supervision and help and counselling, you then have a test. And if indeed Alzheimer's is underway, then you start on existing medication and eventually on the Neuro-Bio medication, which, if the dream comes true, would actually stop any more cells dying, which means if it was taken at the pre-symptomatic stage, then the symptoms would never appear. So that's what we're dreaming to do, and that's what the treatments will involve.
Yes, well, look, that's very exciting. I think that's what most of us would hope that we would eventually get to in this space, because now People, as they get older, in many cases, they're now very well aware of the risk for Alzheimer's disease, and many people have seen family members or close friends suffer the disease, and it's really quite a frightening thing to watch as you see people around you lose their ability to handle their own affairs. So the idea that one would be able to have a way of monitoring the risk for full-blown dementia in a patient-friendly and cost-effective way is tremendously important and very promising. And I think that there should be a lot of support for this. I think we're fortunate that we, as you've mentioned, we live in societies where governments and the people that support those governments have actually devoted some funds to trying to solve these problems. And it's great to see that happening. I would go back to just to encourage you to keep in mind your other potential indications. You mentioned Viagra, and it's not an atypical thing in the drug development world. Viagra was, I think, originally developed as a potential treatment for hypertension because it's a vasodilator, and they thought it would manage hypertension, and it does to some extent. But it wasn't a particularly good antihypertensive, but the fact that it increased blood flow to reproductive organs turned out to be clinically very useful. As you probably know, it also turned out to be quite useful for another disease, pulmonary arterial hypertension, which is a mechanistically similar disease restricting blood flow to the lungs, but it's a very serious and often fatal disease. So, drug development sometimes goes in strange directions. And as you point out, the mechanisms that you're proposing for cell death in Alzheimer's disease, they could apply to cell abnormal proliferation in other diseases. So the idea that you look simultaneously at potential cancer indications actually seems quite sensible to me, and I think opens up other commercial prospects for this approach.
Yeah, so we think what's next for Neuro-Bio, if we can actually develop an effective treatment for Alzheimer's, we wouldn't want to stop there because you can always improve on the medication. And our current prototype drug, which is 14 little building blocks, but gets into the brain, we've got another one in development that's only 6. That's less than half the size. And the smaller a drug is, the more easily it will get into the brain, the cheaper it will be to make. So we are constantly working away at more. It's a bit like starting off with a biplane, but wanting to end up with Concorde. It's good. The biplane's good enough. It will get off the ground. It will do its job. But you still want to devise ever better ways of getting off the ground. Eventually, with something like Concorde, you'd want to do. So that's one of the things we're doing at Neuro-Bio. The other is, and this is going back to my heritage as a basic scientist, is we are looking at this T14 process as a basic biological process and trying to understand how it's made, what limits it, under what conditions it appears. Because the more we do that, the more we can have new targets for Alzheimer's, not just blocking the way it sits at its so-called receptor, but if we knew how it was made and under what situations, perhaps we could intervene at that stage, say. Or we could have an antibody to T14, and we published last year that the antibody to T14 is also a successful blocker of its actions in the test tube, in cell cultures. So we'd like to do that, but as you say, Richard, what's very exciting is exploring applications to metastases. So as you may be aware, that's one of the biggest unmet clinical needs. People die with cancer usually due to the secondary spread, the dissemination, rather than the primary tumor. And there's no, to the best of my knowledge, there's no effective antimetastatic drug. So if one had a biomarker that could tell you metastases was underway and then intercept early on, that would probably be a great step as well in oncology. And then finally, there's dermatology, conditions like eczema and psoriasis, which are due to excessive proliferation of the key cells in the epidermis, so-called keratinocytes. If one had a way of blocking that or suppressing that, that would be very helpful to people with skin disorders. So it's a very wide range of conditions. But I think rather than seeing that as a problem, it's a fantastic thing because it actually shows how wide and how basic this application is and how such a building block of cell growth it might be.
It's quite common in the pharmaceutical drug development business for companies, provided that they have the resources to do it. And it's it's a wonderful sort of problem to have if you have plausible case that can be made for multiple therapeutic applications. Oftentimes, a company, a sponsor, will engage in a phase 2 program that may target multiple indications because you just don't know which one will work out. You have a plausible case why each one of them might work out. And if one has the time and the resources to do it, it's prudent, both from a business and from a clinical point of view, to try multiple therapeutic indications and see which one might work out. And you have that opportunity here. It's a wonderful set of issues to deal with.
It is, and especially having different types of drugs, so different ways of combating, means that you could have a different drug for each disorder, which might be appropriate as well.
Yeah.
Before we end, and we're now drawing sadly to the end, Mike, do you have anything else you'd like to conclude?
Well, apart from the rather obvious in that I think excited is not even close to doing it justice in terms of what I think Neuro-Bio could achieve here. I mean, the fact that we're all working towards a goal that's in life sciences, it's an alignment of the commercial and the societal impact is just wonderful. And again, with the potential impact of Neuro-Bio here, the fact that it's based in the UK, founded in the UK, funded by the UK, I think in terms of the geopolitics as well, it's all very well aligned. So I'm very much looking forward to the next few steps that we're about to make.
Okay, so watch this space, because now sadly we're at the end of this particular episode, and thank you so much for listening to this Neuro-Bio podcast with me, Susan Greenfield, Dr. Richard Mohs and Dr. Mike Zurawski. We hope that you found it enlightening and interesting. And join us next time so we continue the journey to explore with Clotovade and Neuro-Bio our journey and our discoveries for the prospect of a cure for Alzheimer's and for the widespread impact of this treatment on everyday life. Thank you so much for listening, and we'll be with you again soon. Thank you.